Good morning everyone, myself Praveen. Today we are going to discuss nutrition chapter. So in this chapter we are going to discuss detail regarding the nutrition. So by the end we are able to get what is nutrition and what is the use of nutrition in all single cell organisms to multicellular organisms. So first let me know what is nutrition. So what is nutrition? Nutrition is the process by which organisms obtain food and utilize it for energy, growth, repair and maintenance. Simply, nutrition is a process by which organisms from single cell organisms to multicellular organisms, from single cell to multicellular organisms obtaining food, getting the food, and that food is utilized for their energy, for growth, repair, for their energy, growth, repair of the cells and maintenance of the body temperature. So this is known as nutrition. Simply, nutrition is the process by which organisms obtain food and that food is used for their growth, body maintenance and repair of the cells. It is known as a nutrition. So as we are the human beings, we are the warm-blooded animals. So what is mean by warm-blooded animals and cold-blooded animals? Can you say the difference between warm-blooded and cold-blooded animals? So warm-blooded animals means the animals which will have constant body temperature throughout their lifespan. Throughout their lifespan is known as warm-blooded animals. So humans are the example of warm-blooded animals. Cold-blooded animals, the animals which will change their body temperature according to the environment temperatures is known as cold-blooded animals. Simply, the animal changes their body temperature according to the environment is known as cold-blooded animals. Next, nutrition we classified into two categories. One is autotrophic nutrition and heterotrophic nutrition. So nutrition is classified into two categories. Autotrophic nutrition and heterotrophic nutrition. Again, the heterotrophic nutrition is subdivided into holozoic nutrition, parasitic nutrition. So, autotrophic nutrition. What is meant by autotrophic nutrition? Two categories. One is autotrophic nutrition and heterotrophic nutrition. Again, the types of nutrition. Nutrition is divided into two categories. One is autotrophic nutrition and another one is heterotrophic nutrition. So you have to know the difference between what is autotrophic nutrition and what is heterotrophic nutrition. So we'll discuss clearly what is autotrophic nutrition and heterotrophic nutrition. In the nutrition, again the autotrophic nutrition and heterotrophic nutrition. The heterotrophic nutrition is divided into parasitic nutrition, saprophytic nutrition, holozoic nutrition. Again, the heterotrophic nutrition is divided into parasitic nutrition, saprophytic nutrition and holozoic nutrition. So what is parasitic, saprophytic, holozoic? We'll discuss further. So again, the nutrition divided into two types: autotrophic nutrition and heterotrophic nutrition. First, you have to know what is meant by autotrophic nutrition. So autotrophic nutrition, the name itself indicates auto means self, tropic means feed, self preparation, self nourishment is known as autotrophic nutrition. Simply, the organisms that will prepare their own food by themselves is known as autotrophic nutrition. And heterotrophic nutrition is a type of nutrition the organism depend upon other organisms for food. Simply, it is quite opposite to the autotrophic nutrition. Autotrophic nutrition simply prepare their own food in the presence of sunlight, chlorophyll, carbon dioxide and water is known as autotrophic nutrition. So whatever, for example, the autotrophic nutrition example is plants. So if the plant wants to prepare their food, so there should be presence of sunlight, chlorophyll, carbon dioxide and water. These four categories should be present at same time. Then only the process of self preparation will take place. The process of photosynthesis will take place. Only the photosynthesis will take place in the presence of sunlight, chlorophyll, carbon dioxide and water. In the presence of all these categories, the photosynthesis will take place. Next, coming to the heterotrophic nutrition, the organisms depend upon other organisms for food is known as heterotrophic nutrition. The examples of heterotrophic nutrition: all human beings, animals are the examples of heterotrophic nutrition. Examples of autotrophic nutrition: all green plants. So all green plants contain the pigment, green color pigment known as chlorophyll. All green plants present the green color pigment is known as chlorophyll. So given again the heterotrophic nutrition is divided into holozoic nutrition, saprophytic nutrition and parasitic nutrition. First we are able to discuss what is meant by holozoic nutrition. The food is ingested, digested, absorbed, assimilated, digested. Example given as a human being. Simply what is given? Holozoic nutrition is a type of nutrition. The food is ingested. Whatever the food is taking inside by chewing, whatever the food is taking by inside. After whatever the food enter inside, what happen? The digestion will take place by the releasing of some digestive glands, digestive enzymes. After digestion, what happen? Absorb the nutrients and everything. So after digestion, it will absorb the nutrients. After absorption, that assimilation means transport of nutrients through all over the body parts, all over our body. Assimilation, again the undigested food is able to eliminate outside of our body. This is known as the holozoic nutrition, and example is human beings. This is about holozoic nutrition. I will repeat once again. Holozoic nutrition. Whenever, for example, take the human beings. Whenever you were eating the food is entering into your mouth, then it travels to the esophagus. Again, after that it reaches to your stomach. At stomach, the digestion will take place. After digestion, what happens? The digested food will be absorbed. The digested nutrients will be absorbed, and that absorbed nutrients will transport all your body parts through the finger-like projection. At small intestine, there will be finger-like projection is known as villi. From this, the absorbed nutrients will transfer to the blood vessels, and this blood is able to transfer all our body parts. So after that, the remaining undigested food digested, sending sense outside of our body through the excretion. Examples: human beings, and amoeba is also process of holozoic nutrition. Next, coming to the saprophytic nutrition. Saprophytic nutrition. So next to saprophytic nutrition. So organisms feed on dead and decaying matter. Example: fungi. So whenever this type of nutrition, the organisms which will feed on dead and decaying matter is known as saprophytic nutrition. So for example, when you found the dog was dead at an environment, the organisms feed on dead and decaying matter. So simply saprophytic, these are the organisms, these are microscopic organisms. They will take the nutrients from the dead and decomposed organisms is known as saprophytic nutrition. Simply they will feed on the dead organisms, dead materials as well as decaying matter is known as saprophytic nutrition. Example: fungi. 1, 2, 3, start. Next, coming to saprophytic nutrition. Saprophytic nutrition means the organisms feed on dead and decaying matter. The organisms that feed on dead and decaying matter is known as saprophytic nutrition. So these are the microscopic organisms that will get the nutrients from the dead organisms and decaying matter is known as saprophytic nutrition. Simply the organisms which will absorb the nutrients from the dead and decaying matter is known as saprophytic nutrition, and example is fungi. Next, parasitic nutrition. So parasites obtain food from living hosts. Example, Cascota and tapeworm. These are the type of nutrition. These are type of organisms that which will live inside the body or on the surface area of the body and take the nutrition, directly take the nutrition from host material. For example, plants. So take the Cascota plant. Example as a Cascota plant. So in the Cascota plant, it will be a ring spiral-like structure. It will spread to the trees and everything. So whenever the Cascota plant spreading over the tree, the roots of the plant directly penetrate into the xylem and phloem of the plant material. So xylem and phloem of the plant material, from the phloem directly it will absorb the nutrients from the host plant, is known as the parasitic nutrition. And tapeworm, these are also organisms. Look, tape-like structure, tape-like structures. These are tapeworms, and these were able to found in animals in the small intestines. These are about the parasitic nutrition. Next, photosynthesis. Next, photosynthesis. What is meant by photosynthesis? Photosynthesis is a process by which green plants prepare food in the presence of sunlight and chlorophyll. Green plants will prepare their food. Terms and conditions, they will prepare food in the presence of sunlight and chlorophyll, as well as carbon dioxide and water. In the presence of 4 elements, then only the photosynthesis will occur. In the presence of sunlight, chlorophyll, carbon dioxide and water, H2O. In the presence of these elements, the plants are able to prepare their own food. Those are known as autotrophic nutritions. So what are the requirements for photosynthesis? I said you 4 things. Plants will prepare their food in the presence of 4 elements. Those are sunlight, chlorophyll, carbon dioxide, CO2, water, H2O. So these 4 things should be necessary for photosynthesis. For example, chlorophyll is present, sunlight is present, carbon dioxide is present. If the water is not getting, so the process of photosynthesis will not take place. If the carbon dioxide was absent, the remaining things were present, then the process of photosynthesis will not take place. The process of photosynthesis will take place in the presence of 4 elements, that is sunlight, chlorophyll, carbon dioxide, water. Next, after the preparation of photosynthesis, the chlorophyll will prepare during the photosynthesis. After preparation of photosynthesis, the glucose and oxygen are formed. So after preparation of photosynthesis, glucose and oxygen were formed. Then so the process of nutrition occurs in human digestive system. So for humans, the digestion will take place from mouth, esophagus, stomach, small intestine, large intestine, rectum and anus. From mouth to anus, the digestion will start from the mouth and ends with the anus, is known as alimentary canal. So the process of digestion that starts from the mouth and ends with the anus is known as the alimentary canal. So from mouth, what happens in the mouth? When you are taking food, while you are eating the food, so from mouth, while you are taking human digestive system. Human digestive system starts with the mouth, followed by esophagus, stomach, small intestine, large intestine, rectum and anus. The digestion starts from mouth and ends with the anus. The digestion that starts from the mouth and ends with the anus is known as alimentary canal. Whenever the food enters into the mouth, it is mixed with the salivary glands. The food is mixed with the salivary glands. When the food gets entered the mouth, it is breakdown into smaller pieces. It is breakdown into simpler ones with the help of our teeth, upper and lower jaw. The upper and lower jaw helps the food particles to make into simpler, smaller pieces. During that mixing, it is mixed with the salivary glands. It is an enzyme. It is mixed with the salivary glands. During mixing the salivary glands, the food particles will form into round ball-like structure. This round ball-like structure is known as bolus. The round ball-like structure is known as bolus. From here, the food is entered to the esophagus. From esophagus to stomach, the food is traveled through the movement known as peristaltic movement. The food is traveled through the movement is known as peristaltic movement. So whatever the round ball-like structure bolus will enter to the esophagus. From esophagus to stomach, it will travel through the peristaltic movement. Simply, peristaltic movement means the contraction and relaxation of the muscles. The contraction and relaxation of the muscles. During the contraction, the food is pushed to the downwards. After pushing, it is relaxation. So due to the contraction and relaxation of this movement, the food bolus will enter to the stomach. After stomach, it is followed by small intestine, large intestine, rectum and anus. We will discuss further regarding detailed rectum and anus. So we will discuss detailedly in the human digestive system step by step. Next, steps in nutrition: ingestion, digestion, absorption, assimilation and digestion. So steps in nutrition. Nutrition, the small intestine. In the small intestine, the food is able to absorb. The digested nutrients will be absorbed through the finger-like projections. Finger-like projection is known as villi. These villi are connected to the blood vessels. So by this, the nutrients will absorb, and the absorbed nutrients will enter to the blood, and the blood is able to transport to the all over body parts through the villi. The finger-like projection is known as villi. Next, the remaining undigested, unabsorbed food will enter to the large intestine. In the large intestine, the water will absorb and it is entered to the rectum, and remaining undigested food is entered through the outside of our body through the excretion through the anus. Next, steps in nutrition: ingestion, taking food inside. After taking the food inside, what happens? The digestion will take place. After digestion, the digestion nutrients will absorb through the finger-like projection known as villi. After the absorption, assimilation means the absorbed nutrients will transported to all our body parts through the blood vessels. And digestion, the remaining undigested solid waste will be eliminated outside of our body through the anus. Peristaltic movement. What is peristaltic movement? A wave-like muscular movement that pushes food into alimentary canal. So from esophagus to the stomach. From esophagus is a peristaltic movement, a wave-like muscular movement push. So the contraction and relaxation takes place. When the contraction takes place, the food bolus will move forward. So by this process, the food molecules, the bolus food will enter to the small intestine. After it is followed by small intestine, enter to the stomach. It is entered to the stomach, followed by duodenum. It is entered to the stomach and followed by small intestine, large intestine, rectum and anus. So peristaltic movement, a wave-like muscular movement that pushes food into alimentary canal. Next, malnutrition. What is meant by malnutrition? Whatever the nutrients like carbohydrates, proteins, fats, vitamins, minerals. These are the nutrients. Carbohydrates, proteins and fat, these are the macronutrients, and these were micronutrients. Vitamins and minerals are the micronutrients. Macronutrients means the nutrients, the supplements which will need in larger amount, larger quantity is known as macronutrients, and micronutrients, the nutrient supplement which will require in lesser quantity is known as micronutrients. Vitamins and minerals are the micronutrients, and carbohydrates, proteins, fats were the macronutrients. So malnutrition, a lack of protein nutrients in food causes the malnutrition. Whenever you are taking insufficient of nutrients is known as the malnutrition. Due to the malnutrition, you will get some diseases, that is Kawashika. Due to malnutrition, you will get Kawashikar. Kawashikar is a deficiency due to the protein deficiency. When you are not taking proper protein content to your body, then you will suffer with the Kawashikar. Next, marasmus is a disease caused due to protein deficiency and calorie deficiency. It is caused by both protein and calorie. If you are not taking supplement amount of protein and calorie, you will suffer with the marasmus deficiency diseases. So remaining we'll discuss in the next topic. So next, air and photosynthesis. Air and photosynthesis. The scientist Joseph Priestley in the 70th to 80th century, he found that and given the statement that plants are also carrying some amount of gases. And with an experiment, Belger experiment, he given that plants are also giving some amount of oxygen. So with an experiment, want to start. Air and photosynthesis. The scientist Joseph Priestley in the 70th to 80th century, he found that plants are also giving some amount of oxygen, some amount of gases and oxygen. With an experiment, he said that the plants are also providing some amount of oxygen. So this is an experiment. It is a Belger experiment. So in the Belger experiment, he taken the candle, mouse, and a mint plant. In the first step, in the first category, he placed only burning candle inside the Belger. So after 3 to 4 minutes, the candle will be turns off. The reason after 3 to 4 minutes, it is for starting 3 to 4 minutes, it is able to burn. But after 3 to 4 minutes, the candle will turns off. The reason for turning off the candle means before the inverting the Belger, whatever the oxygen presenting inside the Belger, it consumed by the candle. After some time, the oxygen will be completed, then the candle will be turns off. But in the second case, he placed a mouse and candle. In the second situation, he placed both mouse and candle in a Belger. So in this situation also, after some time, the candle will be turned off, and automatically after some time, the rat are also unable to survive, unable to sustain. So in this in the third step, he taken the 3 materials. One is spotted plant, that is spotted mint plant, a mouse and candle. So he kept these 3 in a Belger and leave it for some time. After some time, you are able to observe that the mint plant, mouse and candle he taken 3 materials, that is potted plant, mouse and candle. So after some time, you are able to see that the candle will start glowing continuously. The mouse able to living without getting any suffocated and everything. The plant is also able to living. So by this experiment, by this third situation, we can say that the plant, whatever the plant, it is giving oxygen. The plant leaf is releasing the oxygen. That oxygen is utilized by the candle as well as the mouse. The mint plant is releasing some amount of oxygen. That oxygen is utilized by the candle as well as the mouse. Here the mouse is releasing the carbon dioxide outside, carbon dioxide out. This carbon dioxide is utilized by the plant. By this changing, these 3 materials are able to surviving whole life. This is about Belger-Priestley experiment. So next, activity 2. Activity 2 regarding the carbon dioxide is necessary for photosynthesis. Carbon dioxide, CO2 is necessary for photosynthesis. During the photosynthesis process, the carbon dioxide should be present. Then only the photosynthesis will take place. So during the photosynthesis, there should be available of carbon dioxide, water, sunlight and chlorophyll. If the plant should be prepared, the plant want to prepare the photosynthesis, the carbon dioxide should be present, water should be present, sunlight and chlorophyll. In the presence of these four categories, four factors, the plant is able to prepare the photosynthesis. So what is given activity to given that carbon dioxide is necessary for photosynthesis with an experiment is given. So now we are going to discuss that experiment. So look at the first aim. What is that aim? In the CO2 is necessary for photosynthesis. CO2 is necessary for photosynthesis. Material required. So for doing this experiment, what are the material required? For doing this experiment, what are the material required? Say what are the material required? Potted plant. Material required. So take the potted plant. Next, wide mouthed bottle which is transparent wide mouthed bottle, split cork and KOH, potassium hydroxide. Then you have to take iodine solution. Procedure. So what is the step by step? How to do this experiment? So first you have to take the potted plant. So keep this plant for 2 to 3 days under a dark room. So if you are keeping this plant for 2 to 3 days in a dark room, then this plant will get de-starched. So what's the meaning of de-starched? Simply de-starched means removal of starch from the leaves is known as de-starched plant. So from this for this experiment we have to take the de-starched plant. To get the de-starched plant, I am placing this plant, potted plant under it at dark room for 2 to 3 days. Then I am getting the de-starched plant. So after that de-starched plant, take the potted plant, then place to the de-starched plant. First take the de-starched plant. To the de-starched plant, take one leaf without plucking the leaf. So you should be careful, the leaf should be attached to the plant itself only. So after that you have to place half portion of the leaf inside the white mouthed bottle. Before placing that one, you have to place some amount of potassium hydroxide solution. Potassium hydroxide solution in the white mouthed bottle. Why you are placing the potassium hydroxide? The potassium hydroxide will absorb the carbon dioxide. So when you are adding potassium hydroxide, this will absorb the carbon dioxide. So for that reason, you are placing the potassium hydroxide, then cover half leaf inside the white mouthed bottle and half leaf should be exposed to the outside of the environment. Then you have to close this bottle with the help of the split cork. Split cork means simply the cap is divided into two equal halves. That is known as splitted cork. Simply like this. So you have to close that leaf carefully, tightly with the help of the split cork. Then keep this potted plant for 3 to 4 hours under the sun. 3 to 4 hours under the sun. Then after 3 to 4 hours, simply pluck out this leaf. Remove this leaf carefully. So pluck out this leaf carefully. Then this leaf should be placed under the petri dish and add a few drops of iodine solution. So after adding the iodine solution, you will find that the leaf which is exposed to the outside of the environment, the leaf which is exposed to the outside of the wide mouthed bottle will be having chloro. The leaf, from this we are able to observe that the leaf which is exposed to the outside of the bottle from this position, at outside of this portion, the leaf which is exposed to the outside of the bottle will prepare the starch. Presence of starch means simply the leaf which is covered outside of the bottle, the leaf is able to preparing the starch, chlorophyll and everything. So then plug the leaf carefully after keeping 3 to 4 hours under the sunlight. So both the glass is able to getting these are transparent glass, it is able to getting the sunlight inside the leaf and it outside our environment also. Then after 3 to 4 hours, plug this entire leaf and take it as a separate part. Okay. So after plucking this leaf, observe here carefully, the leaf which is covered at outer portion, while adding the solution, iodine solution, which is turns into bluish black color, the leaf which is exposed to the outside of the bottle will convert into bluish black color. The leaf which is covered inside the wide mouthed bottle will having colorless solution. So by this presence of bluish black color, there is a formation of starch was formed, and there is no colorless, no starch was formed. By this experiment, we can say that carbon dioxide CO2 is necessary for the formation of photosynthesis. So when you are adding the KOH, simply the KOH will absorb the carbon dioxide. By the observation of the carbon dioxide, this inside covered leaf is unable to prepare the photosynthesis. By this activity, we can say that, by experimentally we can say that carbon dioxide is necessary for photosynthesis. This is modes of experiment. This is also most important, frequently asked experiment. And precautions. What are the precautions? What are the safety measures we should take during the experiments? The precautions are: the potted plant should be properly discharged by placing it under a dark room for 3 to 4 days. Then the leaf should be, half of the leaf should be proper exposure to the outside of the environment, and half of the leaf should be tightly packed into the wide mouthed bottle. These are precautions. Lab activity. Experiment to observe the oxygen is evolved during the photosynthesis in the presence of sunlight. So in the previous activity we see that the carbon dioxide CO2 is necessary for photosynthesis. But in this experiment, during the photosynthesis process, the oxygen was evolved in the presence of sunlight. What is given? To observe the oxygen evolved during the photosynthesis. During the photosynthesis process, the oxygen was evolved released by the plant. With an experiment, this is with an hydrilla experiment. So first aim to observe the oxygen was evolved during the photosynthesis. Material requirements. What are the material required? So what are the materials that we are using to do an experiment? That is known as material requirements. The material requirements are, what is given? You have to take the fresh hydrilla plant, funnel, test tube, beaker and water. So what are the material requirements? The material requirements are funnel, hydrilla plant, these are the water, test tube. So procedure, what are the steps to do this experiment? Procedure. First you have to take the beaker, fill the beaker with the water. So take the beaker, fill the beaker with the water, then take the hydrilla plant. That hydrilla plant should be placed inside the funnel. Look here, the hydrilla plant was placed inside the funnel. Then invertedly place that funnel inverted position at the bottom of the beaker. After placing this one, the hydrilla plant is placed inside the funnel. Then you have to, at the bottom of the funnel, at the top of the funnel, you have to invert the test tube. You have to place the inverted test tube to the funnel, connecting to the funnel as shown in the figure. Then place it under the sunlight for 3 to 4 hours. Simultaneously, you have to do arrange one more experiment apparatus. So thus you have to arrange one more beaker, one more funnel, hydrilla plant, test tube and everything. The difference is, in the second process you have to cover, the black paper. Entire test tube and funnel should be covered with the black paper. So I am arranging two experiments. One is without covering the black paper, black strip, and one is which is fully covered with the black strip, that which is covered by the test tube and inverted funnel plant. Simply you have to cover carefully without exposure to the sunlight. Why I am covering the black paper means, whenever the sun rays are falling, the black paper will avoid the sunlight. The sun rays should not fall on the hydrilla plant. So keep these two materials under the sunlight for 3 to 4 hours, then observe. So after 3 to 4 hours, the observation. So what is observation? So after 3 to 4 hours, the funnel which is covered by the black paper, the funnel which is covered by the black paper, there is no changes initial and after the experiment. There is no changes. But the funnel which is not covered by the black paper, when you are keeping under the sunlight for 3 to 4 hours, the sun rays will fall on the hydrilla plant. After falling the sun rays, what happened? The hydrilla plant start preparing the photosynthesis. The main important thing is, during the photosynthesis, the hydrilla plant will release some amount of bubbles. So during the photosynthesis process, whenever the sunlight falls on the hydrilla plant start releasing some amount of bubbles. So this some amount of bubbles will be collected at the top of the test tube. So by these bubbles we can say that 4 hours, the beaker, the funnel which is covered with the black strip, black paper doesn't change initial and final. There is no change what takes place before and after the experiment. But the funnel which is not covered with the black paper, black cover, which is exposed to the sunlight for 3 to 4 hours, whenever the sunlight falls on the hydrilla plant, the hydrilla plant start preparing the photosynthesis. The main thing you have to find is, the main observation is, during the photosynthesis process, whenever sunlight falls on the hydrilla plant start preparing the photosynthesis. During the photosynthesis, the plant start releasing some amount of bubbles. So these bubbles were collected at top of the test tube. So these bubbles were known as the oxygen. Simple, whenever the sunlight falls on the hydrilla plant start preparing the photosynthesis. During the photosynthesis, the hydrilla plant started releasing bubbles. These bubbles are collected at the top of the test tube. So these bubbles are known as the oxygen. So by this experiment we can say that plants will prepare the oxygen during the photosynthesis process in the presence of sunlight. So by this experiment we can conclude that oxygen was evolved during the photosynthesis. And precautions. So after this process, one more thing you have to do, you have to remove this. After this process, you have to remove this test tube carefully by closing the mouth of the test tube with the help of your thumb finger. You have to tightly close, avoiding releasing some amount of oxygen outside of the environment. So you have to remove the test tube carefully by closing the mouth with the help of your finger or your palm. Then after removing the test tube, it is filled with the oxygen molecules and everything. So then you have to bring incense stick or matchstick near the mouth of the test tube. When you are bringing incense stick or matchstick near the mouth of the test tube, the matchstick or incense stick start burning more vigorously when you are placing near the test tube. Because why it is burning more vigorous means due to the reaction of the oxygen. The combustion will take place in the presence of oxygen. So by this experiment we can conclude that oxygen was evolved during the process of photosynthesis under the presence of sunlight. Next, activity. So in this chapter there were 5 experiments. We will discuss one by one detailedly. So activity 1. We given the activity 1 regarding the presence of starch, means a type of carbohydrates that present in a leaf. So to find out whether the starch is present in a leaf or not, so we can find out with an experimentally. So you given experimentally, for that experiment, what is the aim? So aim to find the presence of starch in a green leaf. Next, material required is given. Next, procedure, observation, result. So how to do this experiment? So first you have to take okay. So first you have to arrange the apparatus as shown in the figure. First we have to arrange the apparatus as shown in the figure. First you have to take the tripod stand and you have to place the Bunsen burner. Next you have to place the glass beaker. After placing the glass beaker, you have to fill the glass beaker with 1/3 of the water. Then you have to take the test tube. In the test tube, you have to pour some amount of methylated spirit. You have to pour some amount of methylated spirit in the test tube, not in the beaker. In the beaker, you have to pour the water. In this beaker, you have to fill with the water. In the test tube, you have to fill with the methylated spirit. Then you have to take one fresh green leaf, that which is well exposed to the sun. Why you have to take the green leaf that only which is well exposed to the sun? Because whenever the leaf is well exposed to the sun, the leaf is able to prepare good sufficient amount of starch. So for that, by that leaf we can find out the starch easily by this experiment. So you have to take the leaf which is exposed to the sun, then place the leaf in a methylated spirit. Place the leaf in the methylated spirit and burn it for 15 to 20 minutes. Let it burn it with the help of the Bunsen burner for 15 to 20 minutes. After burning, you have to carefully remove this leaf with the help of the tongs and then place this leaf in a petri dish. Place this leaf in a petri dish. Look here. So this is a petri dish. You have to place the boiled leaf, green leaf. Then after placing that leaf in the petri dish, you have to add the iodine solution. You have to add the iodine drop by drop on the boiled leaf. So while you are adding the iodine solution, after some time the iodine solution, after adding the iodine solution, the leaf will turn into bluish black color. Whatever when you are adding, the leaf will turn into bluish black color. Look here, same once again. After removing carefully with the help of the tongs, you have to place that leaf in a petri dish. This is a petri dish and this is iodine solution. So after placing the boiled leaf in a petri dish, you have to take the iodine solution with the help of the dropper and add few drops, drop by drop to the leaf as shown in the figure. After adding the iodine solution to the leaf drop by drop, after some time you are able to find that the leaf which is exposed to the iodine solution will turn into bluish black color. Which color? Bluish black color. So whenever this turning of bluish black color, this is an indication of presence of starch in the green leaves. So this is about that experiment. So finally in this experiment, the result will be the leaf which will be added to the iodine solution, that leaf will be turned into bluish black color. Bluish black color. So the bluish black color is an indication for presence of starch in the green leaves. Simply starch means during the photosynthesis, preparation of food, the plants will prepare their own food. The food is stored in the excess amount of food. The excess amount of food that which is stored in the leaf. So that leaf is which is stored in the leaf in a form of starch. So this is about that experiment. I think everyone clear this experiment. So now let's look at one more experiment. Activity 3. Sunlight is necessary to form starch in green plant. For the preparation of starch, for the formation of starch, the plants require the sunlight. In the presence of sunlight only, the plants will prepare the starch. Whatever the excess amount of energy, it will be stored in the form of starch. The glucose, the excess amount of glucose that will be stored in the form of starch. So what are the material required? Take a potted plant, black cover paper or black strip, methylated spirit. Next procedure. First, what are the procedure? In the procedure, you have to make the plant as a discharge plant. You know the process how to make a discharge, right? So to make the plant as a discharge plant, you have to place the plant for 3 to 4 days under the dark room. By that the leaves will get discharged. Whatever starch present in the leaf, the starch will be completely removed while placing under the dark room for 3 to 4 days. Then take out this, after discharging plant, take out the discharge plant, cover half of the leaf portion. You have to cover half of the leaf portion. So half of the leaf should be covered with the black strip. Half of the leaf should be covered with the black strip. If you want, you can write the letters, whatever you want. So then place it under the sunlight for 3 to 4 hours. Place it under the sunlight for 3 to 4 hours. During the sunlight, the sun rays will fall on the black paper as well as the plant. So after 3 to 4 hours, remove this black paper and pluck this leaf and make the iodine test. By the iodine test, the paper which is exposed to the sunlight, look here carefully, the leaf, this region and this region, this S letter, will well exposed to the sunlight. Whenever it is exposed to the sunlight, the photosynthesis of process takes place and the chlorophyll was formed. In the presence of chlorophyll, the leaf which is exposed to the sunlight, this region and the bluish region and this estilator, the leaf, it is very exposed to the sunlight. So when it is exposed to the sunlight, the preparation of starch, photosynthesis will take place, and which is covered by the black strip. That which is covered by the black strip, there is no color change. There is a pale color, no color change. So by that we can say that the leaf which is exposed to the sunlight, only the preparation of starch will take place. By this experiment, the covered black paper will not prepare the starch. Simply it is having the pale colorless, palish colorless, simply the which is having colorless, and which is exposed to the sunlight, which is turned into bluish black color. By this experiment we can say that sunlight is necessary to form starch.